Hello everyone. Today we are going to study a very important topic, that is quality control in a clinical laboratory. Quality control is one of the important aspects of total quality management system in a clinical laboratory. And the main emphasis of quality control is to monitor precision and accuracy of performance of analytical methods. Quality control is a new topic for phase 1 MBBS student, and this topic has been recently introduced in the new curriculum, and in practical examination it carries 10 marks. Hence this presentation will cover only the basic and relevant information about quality control, and that too for phase 1 MBBS students. Quality control in clinical laboratory will be discussed in 3 parts. The first part will include quality control basics. In the second part, we are going to discuss about basic QC statistics, and in the third part, we will be discussing about Levy-Jennings chart and the Westgard rules. So let's begin with the part 1, that is laboratory quality control basics. The specific learning objectives of this session are: at the end of this session, students should be able to define the terms quality control, internal quality control and external quality control, accuracy, precision, sensitivity, specificity, explain the significance of quality control in a clinical laboratory, describe the errors in analytical processes in a laboratory, that is systematic errors and random errors, enumerate the causes of errors in a laboratory. So what is quality control? It is a process designed to ensure reliable test results. Reliable means trustworthy. So we are talking in terms of precision, which we are going to discuss later, with the use of controls and standards to ensure accuracy, and reject results when there is evidence of errors. So these 3 words are very important in quality control, that is reliability, which is also called as reproducibility or repeatability, or precision. So the precision, accuracy and the rejection of errors. These things are vital for the quality control in a clinical laboratory. It is a statistical process. Why it is called statistical process? We will discuss this later, because it makes the use of various statistics to see whether the results are accurate and precise, and whether there are errors during the analytical process of in the laboratory. So it is called a statistical process. So this quality control monitor and evaluate the analytical process that produces patient results, and that result should be accurate and reliable. What are the objectives of quality in a laboratory? So clinical biochemical tests are important to support or reject any provisional diagnosis, to monitor the recovery or following the treatment, or to screen the latent disease in apparently healthy individuals, or to detect the toxic effects of treatment. So the analytical results are very important in the modern medicine, and the objectives of quality is to support the provision of high quality healthcare, which in turn reduce the morbidity, mortality and economical loss. And the quality control also ensure credibility of a lab, that the particular lab is giving reliable and accurate result. It generate confidence in lab results, means both the clinicians and patients, this quality in the laboratory system, or the accurate and precise results will generate the confidence in the consumers, which we can say the patients and clinicians. And in addition to that, it also give laboratory staff confidence in their methods. So these are the objectives of quality in a lab. So as we are concerned with the reliable, that is precise and accurate results, how this reliability is ensured? This reliability is ensured with the regular testing of quality control products along with the patient samples, and comparison of quality control results to the specific statistical limit, that is ranges. So during your laboratory visit, you have seen our clinical biochemistry laboratory, and in the clinical biochemistry laboratory, the analytical system comprises of so many factors like maintenance of temperature, technician or personnel doing the test, the instrument itself, the washing conditions, the reagents and control materials, their storage. So all these things make the analytical system potentially unstable. We cannot guarantee that every day the analytical system is stable, because there can be variation in temperature, there can be any error in the instrument hardware or software, or change in the personnel doing the test can also result into the errors. So it these are the causes of potentially unstable system. Now, how to identify the system is unstable? So it is done with the use of stable monitoring system, and this stable monitoring system is a part of internal quality control, and it uses the quality control material, and this stable quality control material are used to identify the instability in the analytical system. As I have said, we use quality control products to monitor the unstable analytical system. So these quality control products we use daily in the laboratory before processing of patient samples. So these quality control products, what are these quality control products? These are patient-like material, and they are ideally made from human serum, urine or spinal fluid, and these control products can be in the liquid form or freeze dried form, that is also called as lyophilized material. A lyophilized material means it is in like a powder form, and before use you have to add diluent to it and make it liquid. So we can get quality control products in like liquid form or freeze dried form. So depending upon the procurement of a laboratory, the both quality control products are available, and both of the quality control products have certain advantages and disadvantages also. And this quality control product is a product which is composed of one or more constituents or analytes of known concentration. Means this quality control product has can have many constituents like it can have glucose, albumin, bilirubin, means all clinical chemistry analytes it can have, and the concentration of that particular analyte present in that quality control material is known to us. It is provided to us by the manufacturer. There are two types of quality control product. One is normal control product and the other is abnormal control product. So the normal control product contains normal levels of the analyte being tested. For example, blood glucose concentration ranges from 70 to 110 milligram per deciliter. So normal control product will have the concentration of analyte within that normal range. So with the help of this normal control product, you can monitor the patient results which are in that normal range. And what is meant by abnormal control product? It contains the analyte at a concentration above or below the normal range of the analyte. Means this abnormal control product, you can say it is like below the normal concentration, and it can have above the normal concentration. So this abnormal control product is able to detect the errors if the patient sample is having the result which is less than normal or the result which is more than normal. So in a clinical laboratory, you can have three types of control: low, normal and high control. But most of the laboratory uses only two controls: normal control and high control. So good laboratory practice requires testing normal and abnormal controls for each test at least daily to monitor the analytical process. Now let us understand some important quality control terminologies which we have been talking, like accuracy and precision. So what is the accuracy? It is closeness of a result to the actual value, which is also called as true value. Means, for example, the plasma glucose concentration is the actual concentration is 100 milligram percent. So it should show the accurate result. The analytical system should show the actual result that is present in the sample of a patient. So that is called as a accuracy. What is precision? It is also called as reproducibility or closeness of values to each other, or repeatability. Means if I am repeating the sample again and again after some time, means for example at 9 a.m. I process the sample, again at 10 a.m. and 12 p.m. and 2 p.m. So if I am repeating the processing of the same sample, the values that I get should be close to each other. Then only I can say that it is precise. They are precise values. There is repeatability is present. And when you can say the values are reproducible, like for example, in one laboratory you are getting plasma glucose concentration 100 milligram per deciliter, and in the other laboratory you are getting 200 milligram per deciliter. So are the result reproducible? No. So means that there might be some error, so the results are not precise. So for the result to be precise, it should possess repeatability as well as reproducibility. What is inaccuracy? It is also called as bias, and it is opposite of accuracy. It is a measure of systematic error present in the analytical system. And imprecision is opposite of precision. It is a measure of dispersion of random errors. We will talk about the errors in the later part of this presentation. So now we are aware about two important terms, that is accuracy and precision. As I have mentioned, the laboratory results should be both accurate and precise. So let's try to understand the accuracy and precision with the help of this picture. So let us consider this as a bull's eye, and this yellow part is our target, and this yellow part is actually in the analytical results term, it is our true value. So now here you can see that all the hits that you can see here is in the bullseye. Means every time you are hitting the target, you are getting the values near to the true value in the yellow circle itself. So you can see that your results are both accurate and all the values are close to each other, means they are precise. So all the results here are both accurate as well as precise. Now let's take a look at this picture. What you can see here, though all your results or all your hits are close to each other, but they are not near your true value. They are not at the center, at the bullseye. So though they are precise, they are not accurate. So this result shows inaccuracy, though precision is there. So it is the indicative of systematic error. Now let us see this third picture. What you can see here, all the targets or all the results that we obtained, they are near, they are like near the true value, means around the true value. Some 3 results are within the bullseye and some are around the bullseye. So we can say the results are quite accurate, but are they precise? No, because they are not close to each other. So here you can say though the results are accurate, they are imprecise. So there is reproducibility error. So in this fourth diagram, what can you see? All the results are like dispersed, they are away from the bullseye. Only one result is at the, 1 or 2 result is near the true value, and all the results are not also close to each other, so they are not precise, and all the results are not around the true value, not around the bullseye. So here the results are inaccurate as well as imprecise. Now after understanding about accuracy and precision, let us try to understand about the sensitivity and specificity. The sensitivity and specificity can be analytical or it can be diagnostic. For example, when you can say that and the particular method is analytically sensitive. So if that assays, the sensitivity is the assay's ability to detect very low concentration of a given substance in a biological specimen. For example, if any specimen is having the particular analyte, for example protein is present, so and it is present in the very low concentration, so the method is known to be sensitive only if it is able to detect that lowest concentration of that particular analyte. So if any method can detect lowest concentration, then you can say it is more sensitive. So the smallest concentration that can be accurately measured by any assay, that is called as analytical sensitivity. Now what is specificity? It refers to the ability of an assay to measure on particular substance rather than others in a sample. A method's ability to measure only the analyte of interest. For example, you have performed the estimation of plasma glucose by GOD-POD method. So this glucose oxidase peroxidase method is specific for the estimation of glucose present in the sample. It does not detect the presence of other reducing substances present in the sample. So you can say that method has the analytical specificity of detecting only glucose in the sample. While if you take an example of Folin-View method, so that is not specific method. Because it is given positive by all the reducing substances present in the sample. So that is not that much specific as compared to, means that is not a specific method, and glucose oxidase peroxidase is a specific method for glucose estimation. So this is the difference between analytical sensitivity and specificity. So what is diagnostic sensitivity? It is the ability of test to correctly identify those with the disease, means the patients who are having the disease, and if any test is able to diagnose that, it is called as sensitivity. Means it shows us the true positivity, that is, it is the indicator of true positive rate. Specificity is the ability of test to correctly identify those without the disease, means if anyone doesn't have disease, if the test is able to identify that, means if it is giving negative result in the patient who don't have the disease, then it is called as the test is specific, because it is the indicator of true negative rate. So this is the difference between diagnostic sensitivity and diagnostic specificity. Now let us talk about two types of quality control. The one is internal quality control and the second is external quality control. External quality control is also called as proficiency testing or external quality assessment. What is the internal quality control? It is a daily process. In this, routine laboratory tests are assayed and evaluated on a daily basis, and this is done by using quality control material. The concentration of analyte in the quality control material is known to us, and quality control material is run before the processing of patient sample, and the result of quality control material that we get, it is cross-checked with the result that we already known to us, and it is done on a daily basis. If internal QC results are within the specified range that is given by the manufacturer, so these results are then used to validate whether the patient's results are reliable or not, and once the test system is validated, patient's results can be used for diagnosis, prognosis or treatment. So this internal quality control is a measure of precision, and by definition, IQC refers to the set of procedures undertaken by the laboratory staff for the continuous and immediate monitoring of laboratory work in order to decide whether the results are reliable enough to be released or not. External quality control, or it is also called as external quality assessment or proficiency testing. It is done by some external outside agency, and it is used to evaluate your lab's performance with reference to other participant labs. Means it is for the inter-laboratory comparisons. For example, if I am getting results of a particular test in my lab, so are they comparable with the results of other lab? If I want to test that, then I have to use the external quality assessment scheme. So this system allows for comparison of laboratory's testing to a source outside the laboratory. So let's try to understand how external quality assessment scheme works. So there is one third outside agency which is called as IQVIA provider. There are some providers like CMC Vellore, and Randall's International Quality Assessment Scheme, BioRad scheme. In our hospital or in our clinical biochemistry laboratory, we use two IQAS scheme, that is CMC Vellore and RECAS. So if we want to see that the results of our laboratory are comparable with the other labs having using the same methods and same instruments, whether there is interlaboratory comparison or not, whether our results are accurate or not, so we get involved in this EQUAS program. So the EQUAS provider send us the external quality control sample. The result or the concentration of analyte is not known to us. We analyze or process the sample as our patient sample at the end of or during the last week of every month, and then we feed the result in the website of EQUAS providers. So the result from all the laboratory goes to EQUAS provider, and then they analyze report, they calculate the mean and various statistic like standard deviation index, target score, and then they send us the report, and based on that report we come to know that whether our tests are accurate or not, whether our IQC program is accurate or not. External quality assessment, that is EQA, refers to a system in which the performance of a laboratory is assessed periodically and retrospectively. Periodically and retrospectively because we process the external quality control sample in the last week of a month, and we send the results to the external agency, and then we get a report and we get to know the IQC status of our laboratory over the period of that particular month, and it is done by independent outside agency, for example BioRad, RECAS or CMC Vellore, to indicate to the laboratory staff whether there may be shortcomings and hence indicates a need for improving or changing IQC procedures. So by the report of EQA, we come to know that whether our results were accurate or not, and then depending upon the results we can change our IQC strategies. Now let's talk about types of errors. There are two types of errors: systematic errors and random errors. Systematic errors, that affect every test in a constant predictable manner. Random errors are due to lack of reproducibility, and these are the errors that affect individual sample in a random and unpredictable manner. What are the causes of systematic errors? The systematic errors are due to the deterioration of instrument light source, gradual accumulation of debris in sample or reagent tubing, if the reagent are deteriorated or if there is aging of reagents, if there is gradual deterioration of control materials, if there is gradual deterioration of incubation chamber temperature in case of enzyme estimation, or if there is gradual deterioration of calibration. So all these can, or there are other various reasons, but these are the most important reasons which results into the systematic errors. Coming to the causes of random errors, if there is improperly mixed or dissolved reagents, if there are air bubbles in reagents and reagent lines, samplings or reagent syringes, if pipette tips are not fitted properly, if there is a clot in pipetter, if the temperature and incubation is unstable, if there is unstable power supply, poor operator technique, improper mixing of process sample, incorrect reconstitution of control material. So all this results into the random errors. So let us summarize the quality control. It is very important for maintaining accuracy and precision in the analytical system, because it is important for producing accurate result for the proper diagnosis and prognosis of patients. Quality control are of two types: internal quality control and external quality control. Internal quality control is carried out with the help of quality control material. This quality control material can be normal and abnormal, and it is a daily process. We have to run the quality control sample daily before analyzing patients' tests. So internal quality control is a process that monitors accuracy and reproducibility of test results by using control materials. It is used for immediate decisions and it monitors daily precision and accuracy of methodologies, instruments, and personnel. While in case of external quality control, there is involvement of third outside agency which send us external quality control sample. The concentration of analyte is not known to us, and it is for the retrospective and periodic assessment of internal quality control. It is also called as proficiency testing, and it is used to compare one's lab results to the other labs and to evaluate IQC program. It maintains long-term accuracy and it ensures the lab is performing to the external standards. These are the references which are used for making this presentation. Thank you.